right, so moving on to 6.3, the nucleus, you can see there are a lot of different structures involved with this particular cell structure. So we're going to start with protein production, and that's kind of an easy way to move through this, what we call the endomembrane system. It's going to start with the control center, the nucleus. The DNA is enclosed in, like I said, that nuclear envelope. It is a double thick membrane, so it kind of folds over itself. Also, we have what is called the nucleolus or the little nucleus in there. We have DNA, which is going to have to be copied if we were to reproduce that cell. The DNA is also copied into messenger RNA that will then leave the nucleus, which is why we need such big openings called nuclear pores in the nuclear envelope for that messenger RNA to get out into the cytoplasm, which is where the actual protein will be made. So we can see here our nucleus and it kind of blew it up. You can see in here is the nucleolus. Notice the pores that are in that double membrane. You can see what it looks like on an electron microscope. We're going to have our DNA molecule in there. And when we make a copy of it, it's called messenger RNA. And then that messenger RNA goes out to the cytoplasm through the nuclear pore. And that is the gist of 6.3. But we're going to keep moving because, like I said, the nucleus is part of what's called the endomembrane system. So the endomembrane system kind of includes the nuclear envelope, and then we're going to go out to the endoplasmic reticulum, to the Golgi. We're going to transport those proteins either out or keep them in the cell. So that's kind of the gist of what the endomembrane system does. So it is really important to make proteins. Animal cells have to make proteins. Even plant cells have to make proteins, I should say. The proteins are what actually keeps you, I guess you could say. It's all your structural support. It has so many different functions, as we've talked about with our biological macromolecules, all of the things that protein does. In the cytoplasm, the messenger RNA is it's going to hook up to a ribosome, right? So it's going to go to the ribosome, okay? The ribosome will then, well, it's unattached, it's free, okay? This is kind of out of order here, I apologize. So the ribosome is going to take the messenger RNA. Ribosomes start off free, meaning that they're just floating around in the cytosol. There are instructions in the first part of that messenger RNA to tell it that it's okay to make that protein in the cytosol, or due to folding restrictions in that, some proteins require a non-polar. So remember, the cytosol is mostly water, so it's polar. They need a non-polar environment in order to fold correctly. So we have these unattached. So if that's the case, then the messenger RNA that is attached to that ribosome will move to the rough endoplasmic reticulum and embed itself in the surface. Now, the ribosomes themselves are actually made in the nucleolus. So again, proteins are processed and folded in the interior of the rough ER. So we have a couple things going on here. We have the mRNA being copied and leaving. We also have our ribosomes, which is an organelle, leaving through the pores, right? So as those proteins, so let's just say we did connect to the rough endoplasmic reticulum, the inside space are called the lumen. The actual folds we will return, well, they tend to be called cisterns or cisternal space. And what's going to happen is in that rough ER, it will make those proteins and package them in vesicles, which they will then bud off. So we can see here that picture with the messenger RNA hooked up to the ribosome. Ribosome attaches itself to the surface of the rough ER, and then we fold the protein and then it buds off in that vesicle to go to its next destination. Under the electron microscope, you can see the reason we call this the rough endoplasmic reticulum is all of this rough surface, which are the ribosomes attached to the surface of the rough ER. So for the endomembrane systems, remember the nucleus contains the DNA. The cell is going to make a copy of the DNA called messenger RNA. The messenger RNA goes out of the nucleus to a ribosome. The ribosome can be attached to the rough ER or free in the cytoplasm. At the ribosome, the messenger RNA is used as a set of instructions to make the protein, right? So in animal cells, protein production, we're going to have those transport vesicles that left the rough endoplasmic reticulum, and they're going to fuse with the Golgi complex. In the Golgi, it's going to modify, sort, and ship proteins to their final destination, which is why we often call the Golgi the distribution center. So here we can say those vesicles that were coming from the rough ER, they're going to hook up to the folds, the cistern, right? We talk about those folds on the Golgi, and it kind of moves that protein through itself as it does those different modifications. And then once it's ready to go, it's going to bud off again. And then, of course, the cell will know whether those vesicles stay in the cell or are going to go out of the cell. And we can see there on the right the electron microscope picture of the Golgi. Once it reaches the Golgi, it's going to have to exit the cell, and it's going to exit in a process called exocytosis. And this is where the vesicle, now remember the vesicle, the outside of it is simply lipids. And so what happens is when they fuse with the cell membrane, it will then they'll kind of open up and then empty those proteins out into the interstitial space. Okay, so we can see here our summary of that. Messenger RNA is making a copy, goes out the pore, hooks up with a ribosome. In this case, the ribosome is told to attach to the rough ER. You can see your making of your protein inside there. We pinch off with a transport vesicle, goes to the Golgi, gets modified, then it fuses with the cell membrane, and it releases its product. There are some other structures associated with this, and the other one is called, one of the other ones is called the smooth endoplasmic reticulum. Now, this one is called the smooth ER because it doesn't have ribosomes on the surface. So when we look at it under the microscope, you can see it's very smooth. It doesn't have those studs from the ribosome. The main jobs for the SER is to make lipids and to detox. It breaks down chemicals that are in the cell that shouldn't be there. We can see that in different types of cells, you're going to have different amounts of different organelles. So, like in gland cells, you'll have a lot of smooth ER because the smooth ER also makes some steroids. Muscle cells, you'll have a ton of it because it also, the smooth ER also stores calcium. You need calcium for muscle contraction. And of course, you're also going to have a ton of it in liver cells because we all know that's one of the main jobs of the liver is to break down toxins like alcohol, medications, right? So all of those things, like I said, so you're going to have a lot of smooth ER in those types of cell tissue, those tissue types. We also have what's called the lysosome. Notice again, we have a lipid bilayer there. Inside is very acidic. So inside here, our pH is very low. With that, the enzymes in there, they require a low pH to be functional. And so what happens is this vesicle, when it binds with food vesicles, it will then break down those biological macromolecules into their monomers so that the cell can use them for their own proteins. Lysosomes will also break down other worn out or defective organelles. So we can see here we have a food vacuole Ooh, actually, this one, I'm sorry. I thought this was a different slide ahead. You have your lysosome, you have your digestive enzymes in there. This poor mitochondria is no longer functional, it's worn out. They will fuse together, and when it does that, those digestive enzymes are then released and it can break down the mitochondria. We will keep the materials that we can recycle. We will export the waste product. All right. So that is it for the two units here, 6364, that I wanted to talk about. I will then move on in the next video for 65 and 66. All right, thanks for watching, and I will see you in the next video. Bye.